Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World, and here we are in our questionnaire session from photosynthesis and higher plants. So yes, today we are going to exercise most of the NEET previous year questions. Okay. So before getting into the questionnaire session, let me tell you in brief about the NEET World special programs. For the 11th standard students, we have NEET World's 2 steps to medicine program, wherein per month the fee is just 3333, okay. And for 12th standard students, you will land on NEET World's 1 step to medicine with the same minimal fee of 3333 per month. And for the long term students or repeaters, you land on the program NEET World's power step to the medicine with the same fee of 3333. This course is for all the students of different financial backgrounds beta. So make use of it to the best possible. So yes, so these courses are not just online, they are online and offline, and complete material will be provided. One-on-one mentorship for the students will be provided with the high expert faculties. And for knowing more details about this course, you can call on to the given number, or you can directly visit the branch which is located in Narayanguda, Hyderabad, and this is the address of our branch. Yes, and the new batch is getting commenced from July 1st, 2026, okay, with the fee of 3333 and with all these facilities. Yes, let's get started with the first question. Look at this, ma. If you know, you can already answer this question also. See, which scientist showed that green substances in the plant chlorophyll is located in special bodies chloroplast within the plant cell. So, in the plant cell, where is the chlorophyll actually located is said by which scientist? It is by Julius von Sachs. He is the scientist who said that glucose is the primary product of photosynthesis. Glucose is the primary product of photosynthesis. Fine. So, this is said by whom? Julius von Sachs. Next. This question you see beta, very simple again. Match column 1 and column 2 and select the correct option from the codes given below. Okay. So, look into the codes once. For C4 plants, immediately what you will remember, ma? C4 plants will show what? Cran's anatomy of bundle sheath cells, right? Of bundle sheath cells. Next you see chlorophyll B. Chlorophyll B will be showing what? The photosystem 2 is linked with what? Photo oxidation of water molecule. This is in the thylakoid lumen. Okay. And here you see chlorophyll B. These are accessory photosynthetic pigments. Tell me few more pigments of the accessory photosynthesis which are called as accessory pigments or antenna pigments along with chlorophyll B. Tell me few more pigments beta. Those include xanthophylls and carotenoids. They land on the accessory pigments or antenna pigments, we will call it as. They will absorb light at different wavelengths which is less than 680 nanometers, and they will get, the energy will get excited, and that energy is submitted to the reaction center. In the reaction center, what molecule is present? Chlorophyll A. Chlorophyll A is present at the reaction center. So, here with respect to chlorophyll B, accessory pigment, chlorophyll B is under accessory pigment or antenna pigment. Photosystem 2, which deals with photo oxidation of water, this happens in thylakoid lumen, wherein single water molecule is photolyzed to 2H plus 2 electrons plus oxygen atom. Clear, ma? Next, if you see CAM plants are what? Succulents or desert plants. Now, you identify the correct statement beta. The correct statement will be 1 is what? 4, 2 will be, yes, 2 and 3 and 4, 4 is 1. So, option A will be the correct answer. With respect to the previous question, I would like to recall the complete discoveries of the photosynthesis by giving you a quick diagram or quick flow chart, wherein from single standard equation of photosynthesis you will be remembering the complete discoveries given in the early experiments. Okay. So, here you see ma, what is the standard equation of photosynthesis? 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll. It will give the primary product of photosynthesis, and water molecules and oxygen is evolved as a byproduct. So, listen carefully beta. Carbon dioxide is essential for photosynthesis is said by Moll's half-leaf experiment. This is said by Moll's half-leaf experiment. Fine. And who said that water and sunlight are essential for photosynthesis? Water and sunlight are essential for photosynthesis is said by Jan Ingenhouse. By what experiment? By hydrilla experiment. And he is the scientist who said that oxygen is evolved from photosynthesis. Make a note beta, oxygen is evolved from photosynthesis is said by Jan Ingenhouse. Clear ma? Next you see glucose. Glucose is the primary product of photosynthesis is said by Julius von Sachs. Julius von Sachs. And who discovered oxygen? Oxygen is discovered by Joseph Priestley. Okay, oxygen discovery is by whom? Joseph Priestley. Fine. By what experiment? Bell-jar experiment. Next. Who said that oxygen is not from the carbon dioxide, but oxygen is evolved as a byproduct from water molecule? That is said by oxygen is from water is said by whom? C.B. Van Neel. C.B. Van Neel. He has conducted what photosynthesis ma? Bacterial photosynthesis. Bacterial photosynthesis. By taking what as an electron donor? H2S. By taking H2S as an electron donor, he has conducted bacterial photosynthesis, and he said that even in oxygenic photosynthesis, oxygen is being evolved from water molecule. Clear, right? Next. So, here carbon dioxide is the primary, next water, sunlight, Jan Ingenhouse by the hydrilla experiment. Glucose is the primary product of photosynthesis by von Sachs. Next, oxygen is evolved from photosynthesis by Jan Ingenhouse. Oxygen discovery is by Joseph Priestley through Bell-jar experiment. Next, by C.B. Van Neel. Clear? He has conducted bacterial photosynthesis, and he said that oxygen is evolved from water. With this, we have finished all the experiments. One thing, who said that? Yes, sunlight is essential for photosynthesis is said by Jan Ingenhouse. But what spectra of sunlight? What spectra of sunlight or what wavelength of sunlight is for the photosynthesis? That is said by E.W. Engelmann. E.W. Engelmann. Okay, he is the person who gave us the absorption spectra and action spectra of photosynthesis. Action spectra of photosynthesis, wherein he said that all the pigments of photosynthesis, all the photosynthetic pigments, be it accessory pigments or the main pigment, core pigment, and accessory pigments or core pigments, what is their absorption maxima, that is being from the absorption spectra. And in action spectra he has clearly stated that which pigment is conducting maximum amount of photosynthesis and maximum oxygen is evolved. Okay, that is being said by what? Action spectra. Clear, ma? So from action spectra he said that chlorophyll A. Okay, chlorophyll A is the main core pigment, fine, that conducts maximum photosynthesis and maximum oxygen is evolved. Clear, ma? Next. Here you see, with reference to factors affecting the rate of photosynthesis, which of the following statements is not correct? Okay, what is not correct he is asking. So here if you see, increasing atmospheric concentration up to 0.005% can enhance carbon dioxide fixation rate. Wrong. The maximum carbon dioxide concentration is 0.05% and the optimum is 0.03%. If it is maximum above than 0.05, carbon dioxide fixation rate will be affected, it will not be enhanced. This we will clearly study in the factors affecting photosynthesis under carbon dioxide concentration. Here you see, ma, next. C3 plants respond to higher temperatures with enhanced photosynthesis, while C4 plants have much lower temperature optimum. This is not correct. Okay. So here if you see clearly, C3 plants will respond to higher temperatures. Wrong. C3 plants will respond to lower temperatures, because these are what? Temperate plants with enhanced photosynthesis, while C4 plants have much low temperature. This is wrong. They will have much higher temperature as they are what? Tropical plants. Fine, ma. So this statement is wrong. Next, option C you see, tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmospheres for higher yield. This is true statement. Next, light saturation for carbon dioxide fixation occurs at 10% of full sunlight. Clear. So this is correct statement. This we will learn in the light intensity factors affecting photosynthesis. So here you see, ma, with reference to the factors affecting photosynthesis, rate of photosynthesis, which of the following statements is not correct? It is obviously option B. Once you look into option A as well, increasing atmospheric CO2 concentration up to 0.05, sorry, if it is up to 0.05, then you can enhance the carbon dioxide fixation. If it is above 0.05, you cannot enhance. So carbon dioxide fixation rate will be affected. If it is up to 0.05, then this statement is correct only. So here the right option is, the incorrect option is option B. Okay. Next question. A plant in your garden avoids photorespiratory losses, has improved water use efficiency. Such high rates of photosynthesis at high temperatures has improved efficiency for nitrogen utilization as well. Have you understood the question, beta? So he is speaking about one plant. That plant is not having photorespiratory activity, means in that plant there is no C2 cycle, and that plant responds in a positive way when there is high temperature. When there is high temperature, there is more amount of photosynthesis. Third is, in the high temperature itself, there is more amount of photosynthesis and there is improved efficiency of nitrogen utilization. Okay. In which of the following physiological groups would you assign this class? Definitely it will be C4 cycle only. Okay. Those are what? C4 plants itself. Because in C4 plants and CAM plants, both the plants photorespiration is not present. But then here he said clearly, nowhere in the question they have mentioned that they are succulent plants, because it is using more water, improving water efficiency is also there in the question. So in that case it will be C4 only. If there is any point like succulent plants, then you can go for CAM. Okay. So this is about this question. Next you see beta. One scientist cultured cladophora in suspension of azotobacter, illuminated the culture by splitting of light through prism. He observed that bacteria accumulated mainly in the region of. So, here you see, okay, what did he do? He has first spread out this unilateral light over the prism. Okay, this is over the prism. When unilateral light is exposed to the prism, this prism will scatter the light to WebGR. This prism will scatter the light to WebGR. Okay, and this scattered light is reflected over the green algae. Okay, this scattered light is reflected over the green filamentous algae, that is cladophora. Over this green filamentous algae, bacteria, aerobic bacteria are placed. Best here he has given acetobacter. So, after a certain period of time, he has observed that near blue light region and red light region, aerobic bacteria got piled up because in this regions maximum oxygen is evolved. I hope you have understood the Engelmann's experiment. What did he do beta? Unilateral light is exposed to the prism and then it got scattered to the WebGR. Then blue light, this when exposed to the cladophora, that is green filamentous algae, which conducts oxygenic photosynthesis. Over this cladophora, aerobic bacteria were placed and this conducts oxygenic photosynthesis. Over the blue light region and red light region, you can see most of the aerobic bacteria got piled up. Okay, you can see most of the aerobic bacteria got piled up because of maximum oxygen evolution. So, with this he said that the bacteria got accumulated mainly at the regions of blue and red light. He said about this. After this he gave us what? Absorption spectra and action spectra. Yes, good afternoon ma, continue with the class. During non-cyclic phosphorylation, when electrons are lost from the reaction center at PS2, what is the source which replaces the electrons? What is the source that replaces the electrons? It is definitely water. How ma? If this is the thylakoid lumen and this is the thylakoid membrane. In the thylakoid membrane or lamellae, here what occurs? Non-cyclic photophosphorylation. Non-cyclic photophosphorylation occurs in thylakoid lamellae or thylakoid membrane. Inside this you will be having what? Thylakoid lumen. In thylakoid lumen what happens? Photolysis of water takes place. Okay. Photolysis of water takes place where water molecule is split into 2H plus 2 electrons plus half O2. Clear? So, this electrons will serve continuously without any interruption of non-cyclic photophosphorylation. Because of this continuous supply of electrons, non-cyclic pathway or this Z scheme is uninterrupted. So, in this case, what is that replacing these electrons and making the cycle run uninterruptedly? That is what? Water. Clear ma? Next. Here you see which of the following statements is incorrect. Rubisco, okay, yes. Which of the following statements is incorrect? Rubisco is a bifunctional enzyme, that is definitely correct because it functions, it binds both to the carbon dioxide and oxygen. In C4 plants, the site of Rubisco activity is in mesophyll cells, wrong. Because site of Rubisco activity where beta? In the bundle sheath cells, not in the mesophyll cells, right. Next, the substrate molecule for Rubisco activity is 5 carbon compound. True. The substrate activity is 5 carbon compound, that is what? RUBP. RUBP is the substrate over here. Rubisco action requires ATP and NADPH. Correct, right? Rubisco activity will require both ATP and NADPH. So, here what is the incorrect statement ma? Statement 2 will be incorrect with respect to C4 plants. Very good. Keep on responding ma, keep on responding to the questions. In light reaction, plastoquinone facilitates the transfer of electrons from, okay, plastoquinone will facilitate transfer of electrons from where to where beta? Photosystem 2 to cytochrome B6F. Let us recall it quickly ma. So, if this is the thylakoid membrane, first to the reaction center, that is P700, and the primary electron acceptor is what? Pheophytin and then plastoquinone. So, from PS2 photosystem 2, plastoquinone will submit to what? Cytochrome B6F and then to plastocyanin. This is the pathway that you will see in what? Photosystem 2. So, here what will be your answer ma? From photosystem 2 to cytochrome B6F, plastoquinone will supply the electrons. You have to remember each and every molecule in this ETS pathway beta, before and after. Find here plastocyanin is the last electron acceptor of PS2, fine, and the primary is pheophytin, remember that. Next, the oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of what? This leads to the formation of one molecule of C3, 3 compound, and one molecule of 2 carbon compound. One molecule is what? 3 carbon compound. What is that beta? Phosphoglyceric acid. One molecule of phosphoglyceric acid plus one molecule of phosphoglycolate is formed. Okay. So, here you see the oxygenation activity, it forms one molecule of 4 carbon, wrong, 2 molecules of 3, wrong, 1 molecule of 3 carbon, correct, 1 molecule of 6, wrong. So, what option we have to go through? Option 3, fine. If in that case 1 molecule of 3 carbon and 1 molecule of 2 carbon also is fine. If there is only one statement which is giving 1 molecule of 2 carbon also, it is fine. Fine, you have to go according to the list of the options given. Fine beta. Next question you see, which of the following statements is incorrect? Okay, which of the following statements is incorrect? Stroma lamellae has PS1 and lack NADP reductase. Stroma lamellae has PS1, that is correct, but it does not lack NADP it lacks NADP reductase, that is true, but NADP will be present because NADP is the ultimate electron acceptor or final electron acceptor of Z scheme. Fine, NADP is present, not NADP reductase. So, this statement is true only. Grana lamellae has both PS1 and PS2. True, grana lamellae will be having both PS1 photosystem 1 and photosystem 2. True. Next, cyclic photophosphorylation involves both PS1 and PS2. Cyclic photophosphorylation, wrong. Cyclic photophosphorylation will involve only PS1, not PS2, because cyclic photophosphorylation is operated in stroma lamellae. Okay, this is operated where beta? In stroma lamellae. Fine ma, both ATP and NADP H plus are synthesized during non-cyclic photophosphorylation, wrong. Yes, they are synthesized, both of them are synthesized, but immediately is NADP H plus. Later through chemiosmotic hypothesis, it is clearly explained how ATP is being synthesized. Both are synthesized through non-cyclic PP. This is correct only. So, what is the incorrect statement over here? It is option 3. Look at here beta, this question, which one of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? Which of the following is not true, he is asking. With regards to what? Chemiosmosis. It involves breakdown of proton gradient, that is true, it is the breakdown of proton gradient, not electron gradient. It is not the breakdown of electron gradient, it is the breakdown of what? Proton gradient. Movement of protons across the membrane to stroma, true. Across what membrane ma? Thylakoid membrane. Next you see reduction of NADP to 2 NADPH on the stroma side of the membrane, correct. So, if you look into this beta, if you consider this as a chloroplast and this is the membrane, from the thylakoid membrane towards the stroma, H plus ions will be accumulated because of the photolysis of water in the lumen. but H plus ions in the stroma will be decreased because of the three things. One is plastoquinone will be submitting the protons to the lumen. Second is through NADPH formation, stroma will get reduction of the H plus ions. That is the reason H plus ions, those are protons, will be less in the stroma and more in the lumen. Fine. As a result, what is generated? Proton gradient is generated from lumen to the stroma. Okay, from lumen to the stroma, proton gradient is generated, not electron gradient. So, what is the incorrect statement here? Option 2 is incorrect, but then in the question not true he asked, right? So, it is option 2. Clear, ma? Next question. Yes. Last two questions beta. Given below are two statements. Statement 1, if you see, the primary carbon dioxide acceptor in C4 plants is PEP, phosphoenol pyruvate, and is found in the mesophyll cells. This is true, correct. Second statement, if you see, mesophyll cells of C4 plants will lack Rubisco enzyme. This is also true. Mesophyll cells will lack Rubisco, bundle sheath cells will have Rubisco. So, both of the statements are correct. So, we have to go with option 1, that is both the statements are true. Next you see ma, this question. What is the role of large bundle sheath cells found around the vascular bundles in C4 plants? What is the role of this large bundle sheath cells which are found around the vascular bundles? To provide the site for photorespiratory activity? Wrong. It is the mechanism to escape the process of photorespiration, right. Next, to increase the number of chloroplasts for operation of Kelvin cycle. True, because so much number of what are present? Agranule chloroplasts are present in the bundle sheath cells. So, if you have to consider this as bundle sheath cell, what are present over this ma? Agranule chloroplast. Agranule chloroplast. Grana is absent, but then stroma lamellae is present. As stroma lamellae is present, it has photosystem 1. Both PS2 is absent because over grana PS2 and PS1 will be there. So, here only stroma lamellae will be there in the bundle sheath cells. So, PS1 only will be there. As a result, you can also add on one point that there is no photolysis of water. There is no photolysis of water. Clear ma? Keep these points in your mind, so much important with respect to bundle sheath cells. So, here to increase the number of chloroplasts for operation of cell cycle, and then to enable the plant to tolerate high temperatures, to protect vascular tissue from high intensity. Look at here, to increase the number of chloroplasts for operation, this is correct. To enable the plant to tolerate high temperatures, nowhere this is responsible for that. To protect the vascular tissues from high intensity, no, because they are already the tropical plants, they have that adaptation. So, what is the correct answer here ma? Option 2 will be the correct answer. With this, today's session will be done, but then tomorrow we are going to exercise most of the advanced questions targeting NEET 2026 with this chapter. So, do not miss tomorrow's session at all, and I would like to close this session by giving you one more time the brief introduction about the NEET World's special programs for 11th, 12th and Power Step. For all the batches, it is the same fee per month, 3333, with all the facilities provided, both online and offline. Complete lectures will be provided with the study materials, okay, with study materials and the questionnaire sessions. And here you see, if you want to know more about these courses, just call on to the given number, or you can directly visit our branch which is in Narayanguda, Hyderabad. Okay. Here you see the new batch is going to start on July 1st. So, do not miss this golden opportunity is what I can suggest for most of the students. Thank you for today, and tomorrow we will meet with advanced set of questionnaire.